Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on histology of the liver. Now, as you can see on the screen, I am showing you one figure, and this figure is of histology of the liver. That the histology of the liver is shown in this figure, and this figure is in two parts. This is the first part that I am showing you, and you go through all the names, try to remember all the names. The reason for showing you this figure that see, in this lecture there are 14 points, and you can remember all this theory point with the help of this figure. I'm showing you the another part of this figure. So, and this is the title of the figure. So with the help of this figure you can prepare this entire lecture, that is the histology of the liver. Now we are starting this presentation lecture. This is the first point. Histology of the liver. Now, histologically, liver is made up of many things. Several components are present in the structure of the liver. Now, which are the structure or components of the liver? That first, hepatocyte. Now, hepat means liver, and cytes means cells. That means the cells of the liver are known as the hepatocytes. And the function of the hepatocyte, that it is performing a metabolic role. Metabolism, we know that there are two kind of processes, that is the catabolism and anabolism. So liver is involved, the cells of the liver hepatocyte, they are involved in the metabolic processes. Then some secretions are performed by hepatocyte, and some hormones are released by the hepatocyte. Now, what are the hepatocyte? First of all, I am showing you in the figure the hepatocyte cell. So here in the figure you can see all these are all hepatocyte. So this reddish brown color cell, all these cells are the cells of the liver hepatocyte. Now, what are the hepatocytes? So hepatocytes are the specialized epithelial cell with 5 to 12 sides, and this hepatocyte they are covering the 80% volume of the liver. So in the structure of liver we can say 80% are the hepatocyte cells are there, and this arrangement of the hepatocyte cell is known as the hepatic lamina. So hepatocyte, they are forming the complex three-dimensional arrangement, and that arrangement is known as the hepatic lamina. So in the figure I am showing you hepatic lamina, so that you will get the idea what is hepatic lamina. But this was the first theory point, and for this you go through this figure, all the names. Now moving on the second point, that the hepatic lamina. Now what are hepatic lamina? So hepatic lamina are plates of hepatocyte, and these plates are having one cell thickness. For example, here this one is the hepatic lamina, this one is hepatic lamina, this one is hepatic lamina, this one is hepatic, this one, this all are hepatic lamina. And as you can see, this hepatic lamina, they are having the thickness of one cell. So right now I am drawing one hepatic lamina. Now this is the another lamina having the thickness of one cell. Okay, this now the third lamina I am drawing. This is the third hepatic lamina. Okay, so these are the hepatic lamina. Now here in this rectangle box, this information is also important. I'm reading this. See, the hepatocyte in the liver lobule form a series of irregular plates arranged like the spoke of a wheel. Okay, here you see plates of the hepatocyte cell, and these plates are the arranged like the spokes of our bicycle ring. So our bicycle ring, the arrangement of spoke, the way the spokes are arranged in the ring of the bicycle, the same way the hepatic laminas are arranged in the liver. Then on the surface, on the top surface, on the exposed surface of hepatocytes, the microvilli are present. So in the figure here, see hepatocyte, I am zooming. So now you can see these are all microvilli. So on the surface of hepatocyte, the microvilli are present. So this was the second point regarding the hepatic lamina. Now in fact this figure is also important, so I am showing you the entire figure. You can go through all the information. All the name at the bottom: interlobular duct, interlobular vein, interlobular artery, then the information regarding the portal triad. You can go through this. Then interlobular septum. Okay. Now moving on the third point. Now third point is regarding the hepatic sinusoid. Now border on either side by the endothelial line, vascular space are hepatic sinusoid. First, what are hepatic sinusoid? Hepatic sinusoid are vascular space, and this spaces, they are surrounded by endothelial cell. So here these are the spaces. These are the spaces, and if you observe carefully and closely, then you will find the endothelial cell. So I am showing you, this is the endothelial cell, this one is the endothelial cell, this one is the endothelial cell. Over here, so in the 3 dimension, you have to imagine how they are arranged. So only the border we are able to see. So this space which is surrounded by this endothelial cell, this space, okay, this space, this vascular space is known as the hepatic sinusoid. Okay. So third point was about the hepatic sinusoid. So here in this figure, hepatic sinusoid, okay, you see, and these are the space, this is the space, and as you can see, it is covered by these spaces are covered by endothelial cell. So these spaces are known as the hepatic sinusoid. Now moving on the fourth point of our discussion. Now the fourth point is regarding the hepatic lamina. Now hepatic lamina are highly branched and irregular structures. In the figure I am showing you, this one is the hepatic lamina, this is the branch of the lamina, this one is the hepatic lamina, lamina, this one is hepatic lamina, okay, and there are so many branches and there is no regular pattern. So this hepatic lamina, they are having the irregular shape, okay. So fourth point, hepatic lamina are highly branched and irregular structures. Now I am showing you the entire figure, so you go through this figure. Then this one is the histological slide, okay. And this figure. Now fifth point. The fifth point is regarding the bile canaliculi. Now what is canaliculi? See, grooves in the cell membranes between neighboring hepatocyte provide space for canaliculi, into which the hepatocytes secrete bile. Here you have to imagine, here this is the hepatocyte cell, this one is the hepatocyte, and this side I am also showing. So this space is the groove, and in the groove this green colored duct is shown, okay. So this green colored duct is bile canaliculi. Now what happens here, the hepatocyte cells are there, and this hepatocyte cell will secrete bile into this canaliculi. So this canaliculi is receiving the bile which is secreted by the hepatocytes. So the fifth point is regarding the bile canaliculi. So I'm showing you this figure. Now, sixth point, what is bile? Now the color of bile, yellow, brownish, olive green liquid, and this bile is secreted by hepatocyte cell. Functions of bile: serves as excretory product as well as also participate in the digestion process. Bile, point 6. Now moving on the 7th point. Now before we discuss the 7th point, I am showing you this figure. Liver structure scheme of liver structure. Try to remember all the names. Okay, all the names. Go through all the names. Then this figure. Okay. Now 7 point. So in the 5th point we have seen that the hepatocyte, they are secreting bile. Bile enter into the canaliculi. Now this canaliculi passes the bile to bile ductules, and this bile ductules join together and they are forming bile duct. So the route of bile: number 1, hepatocyte cell; number 2, canaliculi; number 3, bile ductule; number 4, then bile will reach into bile ducts. Here in this figure, see this I'm saying. See, these are the hepatocyte and this is the groove that we were talking. So in the groove, this green color minute duct is present, and this green color minute duct is known as the bile canaliculi. And with the arrow I am showing you, you observe this, both red color, left and right red color arrow. So you will find this, that the bile canaliculi, they are opening into intralobular bile ductule, and this is a ductule. So canalicules, they are merging with the ductules. Here also periportal bile ductule and bile canaliculi, they are opening into bile ductules, and these bile ductules for example this one, okay, 1 minute, this one, okay, and this one. So this bile ductule, they are opening into the bile duct. So this one is the bile duct and this one is the bile duct. So bile canaliculi open into bile ductule, bile ductules open into the bile ducts. Okay. So this was the 7th point. Now moving on the 8th point. Now what happens to bile duct? So now bile duct is opening into the large right hepatic duct, and some bile duct are going to open into the left hepatic duct. Now you read this point, and we have already discussed this point when we were studying the histology of the pancreas. In that lecture I had explained you, but today in this lecture Also I am explaining this point. With the figure we can easily understand this 8th point. At the end I have kept the figure. We have already seen this figure earlier. See, over here, so bile duct, let's say these are the bile duct, and the bile duct, they are opening into right hepatic duct. So the bile duct of right lobe of liver, they open into right hepatic duct, and the bile duct of left lobe of liver, they open into left hepatic duct. Now right and left hepatic duct, they are joined together and common hepatic duct is formed. So right hepatic duct merge with left hepatic duct and the common hepatic duct is formed. Now here, this is the gallbladder is shown, and this is the duct, cystic duct, and cystic duct is also connecting with this common hepatic duct. So now one duct is formed. So this new duct is known as the common bile duct. Now with this common bile duct, what is happening? The main pancreatic duct. The main pancreatic duct is known as the duct of Versung. So duct of Versung merge with common bile duct, and one ampulla is formed. So we can say in the ampulla, common bile duct and the pancreatic duct, duct of Versung is opening. This ampulla is known as the hepatopancreatic ampulla or the ampulla of Vater. And this ampulla of Vater is opening into this is the duodenum. So here, from the liver, bile is secreted, and from the pancreas, the pancreatic juice is coming. So in the duodenum, we can say the process of digestion will take place, and in that the bile and the pancreatic juice will participate, the enzymes. So this was the 8th point of our discussion. So we are moving back on that point number 8. So this was the point. So if you go through the figure, then you can easily understand this thing. Now, moving on the 9th point. Now, before that, you observe this figure also. Okay. This is the anatomy of liver. Then the 9th point. Now, in 9th point, we are discussing about hepatic sinusoids. Now, before that, I would like to show you the hepatic sinusoid. So, in the figure, here the name is written, hepatic sinusoid, and this one is the hepatic. In fact, all this, the blue color areas, okay, this part, for example, this is the hepatic sinusoid, then this one is the hepatic sinusoid, this one hepatic sinusoid, this one is the hepatic sinusoid, and this rectangle box is the hepatic sinusoid. And now I am zooming this. So here, see, This is figure C, and figure C, a hepatic sinusoid. So you observe this hepatic sinusoid. Okay, hepatic sinusoid. Now, what is this hepatic sinusoid? Now, hepatic sinusoids are highly permeable blood capillaries. Hepatic sinusoids are blood capillaries where they are present between rows of hepatocyte. In between the hepatocyte, blood capillaries are present, and these blood capillaries are known as the hepatic sinusoid. Now, what these hepatic sinusoids are receiving? They are receiving oxygenated blood from the branches of the hepatic artery. So, from the hepatic artery, they are receiving oxygenated blood, and from the branch of hepatic portal vein, these hepatic sinusoids are receiving nutrient rich deoxygenated blood. So for hepatic sinusoid, this thing we are going to remember: hepatic sinusoid are blood capillaries present in between the rows of hepatocyte. They receiving oxygenated blood from branches of hepatic artery, and from the branches of hepatic portal vein they are receiving nutrient rich deoxygenated blood. So this is the 9th point, and this is the hepatic sinusoid. These are all in this figure. All blue color areas are hepatic sinusoid. Okay, this is the central vein in the center. Otherwise, hepatic sinusoids are shown. Now here, this side, hepatic triad. So we are going to discuss about this triad also. Now 10th point. Now 10th point is regarding the hepatic portal vein. Now hepatic portal vein brings venous blood from the gastrointestinal organs and spleen into the liver. So blood is coming in the liver from where? From gastrointestinal organ and from the spleen. And which structure is bringing this blood? So hepatic portal vein is bringing this blood into the liver. Now here you need to understand this thing. See, this is the stomach. This is the stomach. Now the digestion process, there is a digestion in the stomach. Okay, then after the stomach there is a small intestine. The first part, that is the duodenum, then jejunum and ileum. These are the three parts of the small intestine. So in the small intestine also there is a digestion process is going on after our meal. Now when the digestion is completed, carbohydrates, they are converted into glucose. Okay, protein are converted into amino acid, fat are going to converted into fatty acid and glycerol or the alcohol. Okay, DNA, RNA, nucleotide. So these are all the nutrients, and this digested small molecules, they pass the wall of the intestine, they pass the wall of the stomach, they pass the duodenum wall, they pass the jejunum wall, ileum wall. So that is the process of absorption of the nutrients, and these nutrients, they enter into the capillaries, tiny tiny capillaries join together, they form the venules, and venules open into the vein, and this vein is known as the hepatic portal vein. Hepatic portal vein is entering into the liver, and there are many branches of hepatic portal vein. And from the blood, what happens? See here, two things we need to remember. See, digested food material, the nutrients, now they are present in the hepatic portal vein. Hepatic portal vein is reaching in the liver. So some nutrients will enter into the main circulation of our body. See, inferior vena cava is shown over here, and the remaining, which is not required, those nutrients, extra nutrients, will be stored in the liver, and this stored nutrient can be used in future. So nutrients are stored in the liver. So storage function. Now this information, actually this topic, circulatory routes, the hepatic portal circulation. Now you need to read this information, you go through this information. This information is important for you. I am in this lecture not explaining this thing because this all these points, they are from the, this topic is actually of the circulatory system. So when I am explaining the topics of the circulatory system to you, that time I will discuss all this thing with you, but right now you go through this information. And this is the figure. This is also, so hepatic portal system and hepatic portal circulation. This is also, you go through this figure also. Now blood supply of liver. Now all the information you go through, and I am explaining this point. Now see here, hepatic sinusoid. In the purple rectangle box, the name hepatic sinusoid is written. Now two arrows are shown towards the hepatic sinusoid. Here let's discuss this red rectangle box. So in the red rectangle box, oxygenated blood from the hepatic artery. So hepatic sinusoid is receiving oxygenated blood from hepatic artery. Hepatic sinusoid receiving nutrient rich deoxygenated blood from hepatic portal vein. So this is how hepatic sinusoid is filled with the blood, oxygenated or deoxygenated. Now hepatosinusoid, from hepatosinusoid blood enters into the central vein, then blood from central vein enters into the hepatic vein, then from hepatic vein blood enters into the inferior vena cava, and from inferior vena cava blood enters into the right atrium of the heart. So this is the entire route of the blood, okay, and this is the theory point that we have discussed. Point number 11, route of the blood from hepatic sinusoid to heart. Now point number 12, See, blood flows towards central vein. Now, in contrast to blood, the direction of bile flows opposite direction to the blood. Okay. Point number 13. Now, in the hepatic sinusoid, phagocyte cells are present. Okay. In the hepatic sinusoid, phagocytes are present. I'm showing you in the figure. In the beginning, I told you that you observe carefully this figure. See, the reason was this is the hepatic sinusoid, and you see this is this cell, hepatic macrophage. So this hepatic macrophage are the phagocytic cell. Now, what is the role, what is the function of this phagocytic cell? So this macrophage or the phagocytic cell, they are also known as stellate reticuloendothelial cell. So we are remembering this name, stellate reticuloendothelial cell, macrophage or the phagocytic cell. Now what are the function of this hepatic macrophages? So they destroy worn out white and red blood cell. They are killing the bacteria and the other foreign matter in the venous blood, which is drained from gastrointestinal tract. So the unwanted foreign matter from the blood is removed by this phagocyte cells. Okay, so point number 13. Now one more figure, this figure. Now here, this is the final point of this presentation lecture, portal triad. Now what is portal triad? So together bile duct, then branch of the hepatic artery. Branch of the hepatic artery means portal arteriole. Then branch of the hepatic vein, that is portal venule. So three things: bile duct, portal venule and portal arteriole. Together these three items, they are known as portal triad. And as you can see, the portal triad, this portal arteriole with this white color arrow, and portal venule, they are pouring the blood in this hepatic sinusoid. So blood is poured by portal venule and portal arteriole into the hepatic sinusoids. Okay. So this is point number 14, and this was the last point. Now, before I wind up this presentation lecture, I would like to show you the remaining figures. You go through this figure also. This is also important information for you. Okay. In this figure, and see this one. This one is also very important figure. All the names, all the structures you prepare. Okay. Then few histological slide I am showing you. Pig liver, transverse section, primate liver, bovine liver, hepatic liver, lobule, transverse section, bile canaliculi in liver lobule, and this. We have seen this. Then this is the root of bile. And here hepatocyte cell is shown, and this is also very nice figure. See, hepatocyte cells are shown. These are the hepatocyte cell, and bile canaliculi, and bile canaliculi opens into the bile ductule, and bile ductules merge together and forms the bile duct. So you go through all the names. Okay. So with this, we have completed this presentation lecture on the histology of the liver, part 1. Okay, part 2 is remaining. Wait for that. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.